Hello. Do you ever wonder how you developed into the human form and not the shape of a cat or a tube, or even how you're not just a mass of cells? How does everything end up in the right place and at the right length? Developmental biology is the study of this process. In essence, it is the study of how the embryo develops and becomes the adult form. Many theories have tried to explain how the embryo develops. One early theory suggested a very tiny, fully formed person in the sperm just got bigger and bigger on contact with the egg. Amazingly, although we end up very different, many species of animals look remarkably similar at early embryonic time points, particularly in the vertebrates. And it is these similarities and differences that allow us to use model systems such as the frog, chicken, and fly to inform us through experiment on how the embryo develops. A Nobel Prize-winning experiment showed that small groups of cells could instruct others to form a whole new embryo. So it seems that certain cells convey instructions that can inform other cells. These instructions were identified as signaling molecules. One found in fly, known as hedgehog, has a related molecule in chicken known as sonic hedgehog. Yeah, the Sega guy. Sonic hedgehog can induce extra fingers in the chick, showing it to be a signal that can convey instructions for cell fate. Although previously dismissed, the finding that genes and proteins present in flies are also present in vertebrates would open the door for developmental biology. The human condition aniridia, characterized by a small lens, is produced by a defect in the same gene that, when lost in flies, causes a loss of the entire eye, showing that despite roughly 500 million years of evolutionary distance from a common ancestor, the same genes are involved in eye development. Across invertebrate and vertebrate development, it is not new genes for new structures, but a change in where and when genes are used. Look at the spaces between your fingers. These were webbed when you were an embryo, but the cells in these spaces die, creating the gaps. Ducks, however, turn on a gene in the webbed region that prevents cell death. How do these differences in gene expression occur? In regions of DNA previously thought of as junk, various controllers have been discovered that turn genes on or off in cells. It is the evolution and generation of these controllers upstream of genes that can change where a gene is turned on, and thus influence and change development. So the form you take is the product of evolution, gene expression, and cell signaling that has been shaped over millions of years and is only just being understood by developmental biologists. And many questions still remain unanswered. How are these controllers generated, and how do they evolve? And how do the controllers get activated at the correct time in development? Or my own favourite, how do your limbs grow to the same length? And this doesn't even begin to scratch the surface. I mean, what about the plants?